Hello everyone. Plant diseases are caused by plant pathogens, some of which have the ability to modify the plants genetically. Agrobacterium, a soil bacterium which is now known as rhizobium, is one such bacteria which has the ability to genetically modify the plants. In this module, we will study about the two Agrobacterium species, Agrobacterium tumefaciens and Agrobacterium rhizogenes, which infect and genetically transform the plants. The learning objectives of this module are Agrobacterium tumefaciens, TI Plasmid, Agrobacterium rhizogenes, RI Plasmid. Crown gall disease is a common plant disease affecting over 600 non-specified genera or species of plants. The disease mostly affects dicotyledonous species such as woody and herbaceous plants and can be identified by the appearance of tumors or galls of varying size and shape on the lower stem and main roots of the plant. Crown gall disease can affect many commercially important and valuable crops such as grapes, rice and sugar beet. Agrobacterium tumefaciens is a soil-dwelling gram-negative bacterium and is the causative agent of the crown gall disease, an economically important disease of thousands of plants, particularly grapevines, walnuts, apples, horseradish and roses. The ability to cause crown galls, that is the tumorous tissue growths, depend on the ability of Agrobacterium species to transfer bacterial genes into the plant genome. The startling feature is a unique example of inter-kingdom gene transfer. More important to the biotechnologists is the development of plant transformation methods that make use of this unique capability. Plant transformation mediated by the soil plant pathogen Agrobacterium tumefaciens has become the most used method for plant transformation. Agrobacterium tumefaciens naturally infects the wound sites in dicotyledonous plants, causing the formation of crown gall tumors. Since the discovery of the bacterial origin of this neoplastic disease by Smith and Townsend in the year 1907, a large number of researchers have focused on the study of this process, with the hope of understanding the mechanism of oncogenesis in general and applying it to biotechnology. Much research has taken place in an effort to understand the mechanism by which the bacterium infects the plant cells. The bacterium is used as a vector system to transfer foreign DNA into a plant genome with a higher degree of success than is currently seen with other traditional methods. Agrobacterium consists of two main types, those that transfer one tDNA fragment to plant cells and those that transfer two fragments to the plant cells. The bacterium has a circular as well as a linear chromosome and the two plasmid called as AT plasmid and TI plasmid. The pathogenicity of the bacterium depends on the TI, that is the tumor inducing plasmid. The interesting feature of the TI plasmid is the tDNA, the transfer DNA, which is actually transferred from the bacterium to the plant genome. The ability of Agrobacterium tumefaciens to cause crown gall disease depends on the TI plasmid. This is a megaplasmid double-stranded circular DNA, approximately 200 kilobase pairs, present within the bacterium. Analysis of the nuclear DNA in the plant tumors indicated the presence of a part of the bacterial plasmid DNA which was integrated in the host plant. The TI plasmid consists of the following regions: origin of replication, which enables plasmid to replicate independent of bacterial chromosome; plasmid conjugative transfer region, which decides compatibility with other strains; Vir region, which consists of Vir genes; opine catabolism region that breaks down opines to provide nutrition and energy for bacterial proliferation; tDNA region, which is transferred to the host plant cell. The virulence or the Vir region determines the pathogenicity of the bacterium. In response to phenolic signals from wounded plant cells, The Vir-A protein present in the bacterial membrane is autophosphorylated and then transfers the phosphate signal to Vir-G protein. Both Vir-A and Vir-G are constitutively expressed in Agrobacterium. The activated Vir-G binds to the promoters of other Vir genes in the Vir operon. Other than Vir-A and Vir-G that are constitutively expressed, all the other Vir genes are induced when Vir-G binds to their promoters and regulates their transcription. Vir-B gene products are utilized in the formation of the PO complex. Vir-D and Vir-A play a role in generating the transferable form of tDNA, while some Vir gene products are transferred to plant cells along with the tDNA. The tDNA enters the plant cell and is integrated into the plant genome, thus genetically modifying the plant. Details of the transformation process will be dealt with in another module. The tDNA region which is transferred to the host plant is bounded by the left and right borders, which consist of imperfect direct repeat sequences of 25 base pairs. Within the border are genes involved in auxin and cytokinin biosynthesis called oncogenes and genes involved in opine synthesis. The tDNA region is cut at the borders and single-stranded DNA is transferred to the host plant. All the genes in tDNA have eukaryotic promoters since they are expressed in plants. The oncogenes lead to changes in the levels of auxins and cytokinin in transformed cells and cause rapid growth and multiplication of plant cells, leading to tumor formation. The tumor cells synthesize opines. Opines are secondary amine derivatives synthesized from amino acids and sugars. More than 30 opines have been identified in agrobacterial strains, of which octopine, manopine, nopaline and agropine are most common. One of the genes transferred to plant cells is the opine synthase gene. It is expressed in the plant cell and uses plant amino acids and sugars or keto acids as precursors to synthesize these complex amines that cannot be metabolized by plants. The opines are metabolized by Agrobacterium for growth and proliferation. Agrobacterium rhizogenes is a gram-negative soil bacterium that produces hairy root disease in dicotyledonous plants. It induces the formation of proliferative multi-branched adventitious roots at the site of infection, known as hairy roots. The phenolics that ooze out of the wounded plant tissues induce the infection. Subsequently, the genes from the bacterial plasmid are transferred to the plant genome. The virulence plasmid of Agrobacterium rhizogenes is known as the root-inducing RI plasmid, to distinguish it from the tumor-inducing, that is TI plasmid. The RI plasmid shares extensive functional homology with the TI plasmid. The RI plasmid, like the latter, contains a distinct segment of DNA which is transferred to the plant genome during infection. The root-inducing plasmids are large, approximately 200 kilobase pairs in size, and contain one or two regions of tDNA and a virulence region. The RI plasmids are grouped into two main classes according to the opines synthesized by hairy roots. First, agropine type strain induce roots to synthesize agropine and the related acids. Second, manopine type strains which induce the roots to produce manopine and the related acids. The agropine type RI plasmids are very similar as a group and a quite distinct group from the manopine type plasmids. The tDNA region of the RI plasmid consists of two segments. The left segment called TL, which has several genes which include the role A, B, C, D genes that are important for the hairy root phenotype, but whose precise functions are not yet well understood. The right segment is called TR, which also has many genes which include the auxin biosynthesis genes AUX1, AUX2 as well as the opine synthesis gene. Both TL and TR regions are transferred to plant cells and integrate into the plant genome. The module can be summarized as: two Agrobacterium species can genetically transform plants by introducing DNA that enters and integrates into the plant genome. The bacteria have plasmids, which are known as Ti or Ri plasmids, and carry the pathogenicity genes. Virulence or pathogenicity of these bacteria is determined by the vir operon that is induced in the presence of phenolic signals from wounded plant cells. The tDNA from these plasmids is transferred to the plant cell and it carries genes that code for enzymes which play a role in plant hormone biosynthesis. The change in hormone levels cause tumor or hairy root formation. The tDNA also carries a gene encoding for an enzyme that leads to synthesis of specific metabolites called opines. The opines can only be metabolized by the bacterial cells.